Let's now go on with an overview of genetic disorders. This is part one. A genetic disorder is a disease that is caused by an abnormality in an individual's DNA. And by what we mean by abnormality is you can have a mutation in DNA itself. Let's say it changes at one point or the other, or there is an addition or subtraction of an entire chromosome or sets of chromosomes. That is what we refer to as a genetic disorder. Now, these genetic disorders, most of them are very much rare, and they'll just affect one person in a lot of thousands and millions. Genetic disorders may result by, so what can cause genetic disorders? You can have a point mutation. Let's say, for example, in a DNA strand, you're supposed to have adenine. Now you end up having guanine. That is a point mutation. There is a changing or a switching of a base, nitrogenous base. You can also have an insertion or a deletion entirely inside one gene. So maybe you're supposed to have GCCG, but then you just have GC. So there is a deletion, or you can have an insertion. You add other nitrogenous bases on a strand. So that can cause a genetic disorder. You can also have a deletion of a gene or genes that can also cause a genetic disorder. You can also have a O chromosome or extra chromosome missing or both. Okay, you can have an extra chromosome, missing chromosome, or both missing and extra. So anything like that can cause a genetic disorder. So types of inherited diseases, we have got autosomal disorders. Now, autosomal disorders meaning that they affect our somatic cells, not sex cells, but body cells, and they can be autosomal dominant or autosomal recessive. So if you are talking about autosomal dominant, it means that if any one of your parents have them, definitely you are going to have it also. Autosomal recessive, if your one of your parents have it, you will not have it. But if both of your parents have it, you have got a chance of also having it. Or if both your parents are carriers, there's a chance that one child is going to have it. So those are autosomal disorders, and the example of autosomal disorders include Down syndrome, hemophilia, sickle cell anemia. You also have allosomal disorders. Allosomal disorders, these ones are going to be uh, linked to the sex. It can be X-linked dominant or X-linked recessive. Now, Y-linked, we don't have anything to do with Y-linked dominant or recessive, because if it is Y-linked, immediately you have it because we only have one Y chromosome. And the examples of these disorders which affect the sex chromosomes, they include Klinefelter syndrome and Turner syndrome. You also have mitochondrial disorders also, okay, to affect like the mitochondria. Genetic disorders can be classified as single gene disorders, chromosomal disorders, or genetic disorders, or multifactorial genetic disorders. So these are the three classes: single gene, meaning affecting one gene, or chromosomal affecting a chromosome, or multifactorial gene disorder, which is also known as a genetic disorder. Autosomal disorders and allosomal disorders, to compare them, autosomal ones. These ones are going to arise by gene mutation in autosomal chromosomes. And then allosomal, this one will arise when there is a mutation in sex chromosomes, and it's mainly the X chromosome. Y is rare. When we say autosomal chromosomes, we're talking about body chromosomes other than sex. The autosomal disorders, they affect the both sexes, can be in males or females, while allosomal, they affect only one sex, and they are more common in males, okay, than females. That is because the male has got XY. Let's say, for example, we're talking about hemophilia. Immediately you have the gene for hemophilia as a male, it means that you are going to have the disease, okay. That is because the Y chromosome cannot carry that disease. But for a female, they can have the gene for the same disease, let's say sickle cell. They have the gene for that disease, but then the other X chromosome does not have the gene for that disease. So they'll just end up being a carrier. So because women or ladies have got a chance of being carriers, that's why they are less affected by allosomal disorders. Autosomal disorders, again, the mutated gene can be dominant or recessive, while allosomal disorders, the mutated gene is recessive. And then autosomal disorders, the the suffer is homozygous. The one who is suffering is homozygous or heterozygous. Meaning you can have things like Down syndrome, sickle cell anemia, can either be homozygous for the condition or heterozygous. Now for allosomal, the one who is suffering is going to be hemizygous. For example, Klinefelter syndrome. Okay. How are you going to be able to diagnose genetic disorders? How are you going to know that someone has got a genetic disorder? A personal medical history can actually show, is one of the things that you can use for diagnosis to identify that someone has medical history and also family medical history. Physical examination, you see and look at some traits, they can be able to tell. And also investigations, which can include laboratory tests and also imaging. Those can also be used to identify or to diagnose a genetic disorder. Laboratory tests, like the genetic testing, we are going to have molecular, chromosomal, and biochemical genetic testings to be used to diagnose genetic disorders. What is the purpose of genetic testing? You want to finding out genetic diseases in unborn babies. So what will happen is that you are going to be able to test the parents. So the parents are going to go for genetic testing to be able to find out a genetic disease in unborn babies. Okay, what will happen is that you can use the ultrasound and everything like that. Genetic testing is also very important in that you are going to be able to find out if people carry a gene for a disease and might pass it to their children. Because before you can marry that your lover or something, at least you need to make sure you know about the genetic makeup. I was watching a documentary of this lady who was crying because the parents were both having this disease. Actually, they were both carriers. So they didn't know, because they were just lovers, they couldn't go for genetic testing. When the child was born, the child was in great pain the rest of her years. So that is really bad, putting someone in trouble for something that they did not know. So screening embryos for disease is also one of the purposes of genetic testing. So in case the embryo has got a disease, at least if you can detect it early, you can actually do some treatment early or anything that you're able to perform to be able to help. Genetic testing is also necessary for testing for genetic diseases in adults before they cause symptoms. Before symptoms can actually come out, you test the adults. And also making a diagnosis, okay, making a diagnosis in a person who has disease symptoms. You already seen some symptoms, but you want to confirm really if it is this kind of disease, so you get to perform a genetic testing. So these are some purposes. So let's now look at the single gene disorder. Single gene disorder, an example of it is sickle cell anemia. The specific gene mutation that results in sickle hemoglobin involves the substitution of thymine for adenine. So instead of having GAG, you're having GTG. You can see the adenine has been replaced, okay, by thymine. So this is on the sixth codon of the genetic sequence, on the sixth position. And this is going to lead to the coding of valine rather than glutamate, because GAT, this codon, is supposed to give you the amino acid valine, but GTG gives you the amino acid glutamate. So on the sixth position, instead of having valine, you're having glutamate in the hemoglobin beta chain. So sickle hemoglobin, the result of this mutation as a singular property of polymerization when deoxygenated. So in other words, there's no enough capacity of this hemoglobin which is mutated to be able to carry that oxygen. All right. So what are going to be some clinical features of sickle cell anemia? You can see infarction, pneumonia, chest syndrome, you have hypertension, heart failure. So it is really a bad condition. You can see also hemolysis. The red blood cells can actually easily get to break, rupture like that, and that leads to anemia, which is you don't have enough red blood cell count in the body because they are rupturing. Okay, firstly, they had to lose their shape. Because they lost their shape as they are moving in the blood vessels, they can easily rupture because the capillaries are small. Okay, and that is going to lead to some avascular necrosis. You are going to see necrosis happening, tissue being tissue dying, especially on the hip, abdominal pains. Okay, there's also going to be hand foot syndrome, pain. So a lot of conditions, chronic ulcers. So all these are some physical manifestations and some things that you'll be able to identify in someone that has got sickle cell anemia, and also splenomegaly. Okay, the spleen gets to enlarge. You know that the spleen is the one which removes dead red blood cells, and here we are saying there's hemolysis, meaning that the red blood cells are dying, so the spleen has to enlarge for it to be able to contain all those dying red blood cells. Let's now talk about trisomies. A trisomy is a type of polysomy in which there are three instances of a particular chromosome instead of two normal ones. You have got the three chromosomes. It is actually a type of aneuploid. Aneuploid is an abnormal number of chromosomes. If the chromosome pairs fail to separate properly during cell division, the egg or sperm may end up with a second copy of one of the chromosomes. Normally, say this is a cell, you have got a cell. During meiosis, this cell is supposed to divide, and one chromosome is going to go in one cell, the other chromosome is going to go in the other cell. But in case they fail to divide, meaning that both chromosomes will go in the same cell, and that cell forms a sperm, and that sperm goes and fertilizes an ovum which has got one chromosome, you are going to end up with three chromosomes, and that is a trisomy. Okay, the most common types of trisomies that survive up to birth, we are going to have trisomy 21, which is Down syndrome, trisomy 18, that is Edwards syndrome, trisomy 13, that is Petal syndrome. So you can use DEP 21, 18, 13. Trisomy 21, Down syndrome, is going to be caused by it's a condition that is caused by the presence of all or part of an extra 21 chromosome. It doesn't need to be all, it can actually even be just part. Then you have got a 3. Mosaic Down syndrome. So in this rare form of Down syndrome, children will have some cells with an extra copy of chromosome 21. Okay, an extra copy, as you can see here. This mosaic of normal and abnormal cells is caused by abnormal cell division that was happening during after fertilization. So after fertilization, the cells could not divide, and so you ended up with having a trisomy. Translocation Down syndrome. So one is Down syndrome can also occur when part of chromosome 21 becomes attached or translocated onto another chromosome before or at conception. Mosaic Down syndrome, this one is a rare form where children have got an extra copy of chromosome 21, which is caused by abnormal cell at after cell division or after fertilization. Okay, there is an abnormality in division after fertilization. While translocation Down syndrome occurs when part of the 21st chromosome becomes attached, that is to be translocated onto another chromosome before or at conception, at birth, not at fertilization. So what you'll be able to see is that these children have usually two copies of chromosome 21, but they also have additional material from chromosomic attached to the translocated chromosome, to the chromosome that is attached to the 21st chromosome. Okay. So when you are identifying these conditions, these trisomies, you use a karyotype. So this is what we call a karyotype. You want to see how many chromosomes are there on each pair. What are some physical features of Down syndrome? You are going to see a broad, flat face, slanting eyes. You also see a short nose, short hands, and broad. Okay, short and broad hands. They are short, but then they are broad. Small and arched palate. You can see the palate there. Big, wrinkled tongue, dental anomaly, congenital heart disease. Mostly these genetic disorders are going to be followed by congenital heart disease, because as these conditions are happening, they actually affect the way the heart gets to form. And so there's going to also be a lot of problems with the heart. In Down syndrome, you're also going to see enlarged colon. So the large intestines are also large. The big toe, the space between the big toe and the other toes is very big also. You are also going to see a single palmar crease in case of a newborn. This line that is in the hand will just be alone. Normally it's supposed to be somewhere like three, but you only see one. There can be also umbilical hernias where the intestines are coming out through the umbilicus or in the abdomen. There's also an abnormal pelvis, diminished muscle tone. The muscles lack that tone. So a lot of things. And mostly the one feature also that to be able to follow these conditions is mental retardation. The person doesn't look normal, doesn't behave normal like that. Okay, so those are some of the clinical features. And then trisomy 18, Edwards syndrome. Here you can see, if you do a karyotype, you can see that at position number 18, you are going to have three chromosomes, also known as trisomy E. Okay, Edwards syndrome, also known as trisomy E, that E from the Edward. It is a genetic disorder caused by the presence of or an extraordinary, or of an extra 18th chromosome, and that causes that trisomy. What is the cause? There was a meiotic non-disjunction when the cells were undergoing meiosis. The cell did not manage to separate, so you had two chromosomes in a cell which was supposed to have one chromosome, because normally a sex chromosome is supposed to have a haploid number, but it had a diploid number. So after fertilization, you ended up having an extra chromosome. Edwards syndrome can occur in around 1 in 6,000 live births, and around 60% of all those affected being females. So this one is actually more common in females than it is in males. Just something to know I didn't mention, the Down syndrome is the most common type of trisomy. That is because for Down syndrome, those who are born have got a higher chance of surviving than these other conditions. They mostly die before birth or at birth. That is why the most surviving trisomies are those with the Down syndrome. Features of Edward syndrome. You are going to see back part of the skull is prominent. This one, there is a prominent back part of the skull. Also dysplastic or malformed ears, clenched hands with overlapping fingers. You can see the hands like that. Small mouth, small jaw. You can see the jaw down here, very small, and a short neck, almost as though someone doesn't have a neck. So the head is just lying on the neck. I mean on the chest. That is also how you are going to see mostly with those with trisomy 21. It's just that for them, they've got a webbed neck. It's just like the whole head is just connected entirely to the chest. Okay. You're also going to be able to see a shield chest. You can see how the chest is. Okay, that's a shield chest. And short and prominent sternum. The sternum is prominent. You can actually tell, and it's short. And there's a wide distance between the nipples. So there's a distance between the nipples there. Okay, there's a big distance. You're also going to see flexed big toe and prominent heels. You can see the heel is so prominent, and then the big toe is actually flexed. It's bent there. And then trisomy 13, which is the Petal syndrome, also known as trisomy D. Remember, Edward was trisomy E. Okay, this is trisomy D. It's a chromosomal abnormality in which you are going to have an extra of the 13th chromosome. If you come on the karyotype here, you'll be able to see the 13th chromosome, an extra chromosome on the 13th pair. Some of these are going to be caused by Robertsonian translocation. So there's a translocation where a chromosome add actually to the 13th chromosome sticks or is translocated and it has got an extra one like that. The extra chromosome 13 is going to disrupt the normal course of development, and that will cause heart and kidney defects amongst other characteristics of Petau's syndrome. So one common thing that you'll be able to see is a cleft palate. So you can see a big cleft here. Okay, so some common features: mental deficiency. Almost all these conditions, all these trisomies, there's going to be a mental retardation. Low birth weight, abnormal development of the frontal lobe, absence of corpus callosum, hypoplasia of cerebellum, sloping forehead, scalp defects, malformed ears, congenital heart defect, renal tract anomaly, microphthalmia, okay, bilateral cleft, and these other conditions. So you can also see polydactyly. You'll see an extra digit. Instead of having 5, you're going to see 6 digits. Also have Klinefelter syndrome. Now, Klinefelter syndrome is a genetic condition because when someone has two X chromosomes and Y chromosome, so it is XXY. Okay, XXY is going to be caused by non-disjunction during meiosis also, because people with XXY chromosome have a Y chromosome. These are going to be considered as males. Most XXY individuals develop as males, often not knowing they have an extra chromosome. They don't know. So the XXY chromosome arrangement affects primary sexual development. That is one common thing because you have got an extra X. So instead of being a male, normally you start looking as though a female. Typically, tests don't fully develop, and then the levels of testosterone are going to be much lower than average. And this is the most important hormone in males, but they're going to be lower. Someone looks as though they are females. As adults, nearly all XXY males are unable to make sperms, and so they cannot have biological children. Many men discover their condition only after they seek medical help of infertility. That's when they are going to know that they have got this condition, the Klinefelter's condition. Changes that appear at puberty can include low growth of facial and body hair. Now, we're not saying that if you just see facial hair, no, on a man, it means that they have got Klinefelter syndrome. You're also going to see development of breasts or breast tissue and also small testicles. For Turner syndrome is caused by a missing or incomplete X chromosome. So instead of having XX for females, they only have one X. Okay. So the genes affected are involved in growth and sexual development, which is why girls with the disorder are shorter than normal and have abnormal sexual characteristics. So you can see this is how they're going to be able to look. Of course, congenital heart disease also there. You can also see low set ears, droopy eyelids, low hairline, short chest, widely spaced nipples, okay, like that. Short fourth, metacarpal, elbow deformity, pube- pubertal failure. There's failure of puberty there, okay. So, like there is a streak ovaries, amenorrhagia, meaning someone doesn't have menses every month, and there's also going to be infertility. You can see also the kidney, horseshoe kidney, meaning that there's also going to be problems with the renal system. Okay, so these are some things that you'll be able to see. A spaced chest also. So these are some physical manifestations which you'll be able to see. So that is it for overview of genetic disorders, and that is it for the topic. Thank you so much for being present.